Okay, I guess we'll go ahead and get started, you guys. My name is Andy Duncan. I'm one of the site directors here of our pathobiology site, and this is.
I'm Dave Gow. I'm the second site director for pathobiology.
So we're super excited to have everybody here and to really celebrate the whole summer of research that the 7 pathobiology students have done. I think you're going to be really amazed by everything that they've done. And there is so much depth and diversity with what they've spent their time doing this summer that it's just, I'm just really floored. We've been watching them learn and talk about their research all summer, every Friday. And it's just been amazing to see it get better and better every week. And last night I was looking at all the slides as they came in, and it was just like exponentially better and cohesive and just so impressive. So what I want to do is just walk through a little bit of things that are going on and what we've done this summer. So this is us. So Dave and I are the co-directors of this site. This is our first year that we've had this site called Pathobiology. So the Hillman Academy takes around 60 to 80 students every year, and they're arranged geographically. And this is the first time that we've had one here in this building, these couple of buildings here on the Oakland campus, really housed in the Department of Pathology. So Dave and I have been running this. We've had an education team who've done some events. That was Alyssa Armanin and Kaylee Montanari. So they've worked with the students and done some professional development activities. And these are the sites. So we're right here, and this is all of like Pittsburgh. This is the main medical center. There are a couple of sites, I think, that traditionally have had some labs here. One is called Cobra, one is tumor immunology. This is our pathobiology site. There's one down here by the river. There's a vision site over at Mercy Hospital. There's biomedical informatics. And then this afternoon, we'll be at Hillman and the Assembly Building. But we're right here in the heart of Pittsburgh and the heart of the Oakland campus for pathobiology. And this is really tied with our Department of Pathology. And you'll learn more about this as we go on. So I think one of the really cool things about our site in pathobiology is we really highlight research diversity. So as much as I love the sites that do tumor immunology or cancer biology, we're really more eclectic, I think. So we think a lot, the different labs here, about basic and applied research. We do pathology, cell biology. You're going to hear a lot about liver today, some transplantation, neuroscience, lung, kidney, stem cells, and so on. So we touch a lot of different areas. And will you say, how does that relate to Hillman Cancer Center? And I'll tell you, it's because we think a lot about basic and applied research. And if we can understand the mechanisms that sort of regulate biology, we can maybe figure out how to exploit them. So in other words, if you can figure out how things go right, you can then understand how they go wrong. And then can you then adapt that to cancer and other aspects of health and disease? So these are the students, and I just want to introduce them very briefly, and you're going to hear more from them as we go along. We took this picture on the very first day, and you can see they've all, aged and grown wiser, a few more gray hairs. But I want to just really acknowledge 2 of the students first. So Eric and Ezra, these guys, this is their second year. Did I forget anybody? Has anybody else done a second? Oh my gosh, Brandon, you're right. Sorry, I knew that. Okay, sorry, Brandon. I knew that something was wrong. So put a little arrow to Brandon's head right here too. And then we've got the new scholars. So Brandon is a returner. Yeah. Oh, look at that. I forgot to, okay, I made it. Sorry. 4 color schemes. Najib, Alberto, Elise, and Kai are all the new students. And so I think it's been nice having a mixture of people who know how the program works and the new students. And I think this is a really impressive part of the slide. You can start to get a sense for that research diversity that everybody's been working on. So you can just look at the bolded words here. So Eric has worked on liver cancer. Brandon's worked on induced pluripotent stem cells. Najib's worked on acetaminophen or Tylenol overdose. Alberto worked on liver diseases. Elise worked on ALS. Ezra has worked on lung disease. And Kai has worked on endocytosis. So lots of different areas. And how do these all connect to health and disease? That's what you're going to learn about today. None of this could be done by itself. And so everybody, all the students have been paired with laboratories. And so we have these 4 labs that are in this building on the 4th floor. Dr. Vargas's lab is on the 3rd floor of this building. And we've got 2 other labs that are 1 building over. So thank you very much to these mentors. They volunteer out of the kindness of their hearts to do this. You know, they don't get any extra money for this. They just really are passionate about training students. But even then, it's not just the mentors. There's a whole community that supports these students. And so within every lab, we listed here 1 mentor. So for example, in Dr. Dressman's lab, Dr. Kolarchik's lab, Brianna Sullivan was sort of the primary mentor, but in addition to that, there were other people who were helping to support her. And that was the same in all these different labs. In addition to the lab mentors and the PIs, there were people who participated in our seminar series. There were people who worked behind the scenes. There were offices. So, you know, well over 50 people have really dedicated their summer to helping these 7 students. And so I just really want to thank all of those people right now, and we'll give them a round of applause. So what have they done all summer? We had this very complicated calendar right here, and you can see they were here for 7 weeks. If you really squint your eyes and look at that carefully, you'll see there are a bunch of recurring events. So every Monday, we did something called Path Forward, which was really a professional development series that was run by a couple of graduate students. And it talked about, you know, how did certain people get to where they are today? What choices did they make? What accidents happened? Where is serendipity and all that? So hopefully they learned a lot about, the students learned about graduate school and medical school and how to become a professor and things like that. On Wednesdays, they went to the assembly building where we'll be this afternoon and learn some more professional development and science related things. And on Fridays, everybody gave about a 5-minute presentation, and they talked about what did they do this week, what are they going to do next week. And I think that's really one of the best things we do at this site is to get people to talk about their research. So if you can talk about it and you sound like a scientist, you're getting close to being a scientist if you can really talk about it and articulate those ideas. So this is what they did. You guys saw all the sort of streaming pictures as you walked in, but they spent the bulk of their time engaged in the lab doing research, you know, under the guidance of their mentors and labs. And so you can see all these different activities they were doing. Here's some more. And then in addition to, you know, being in the lab, like I said, they learned to talk about the research. So they learned to present their data, to think through it, and then to even watch it and ask each other questions. Which brings me to another point. We're going to see some talks today. You guys are encouraged to ask questions. Anybody can ask a question at the end. We have a few minutes dedicated for everybody. So this is our last slide. And I just wanted, and there are some printouts if you guys don't have this. We've already hit point number 2 here, which is the welcome. And then we're going to jump into the presentations. So everybody has about 15 minutes budgeted. So we asked the students to have about a 10-minute talk. So then they can get a few minutes of questions. Their mentors are going to introduce them. We'll have a little break and then do the final 4 talks. There'll be lunch. It'll get set up right outside where the breakfast stuff is, and we can just eat that right in here. And then for those of you who are going to go to the assembly building for the closing ceremony, that starts at 2. If anybody needs to ride the shuttle, I saw that it's supposed to come on Lothrop, which is right over here, at 12:55. So I think we'll be fine, but I don't know if anybody needs the bus this time or not. So with that, I'm going to turn it over to Dave, and then we'll start.
All right. So just wanted to echo what was said. It's pretty much, it's always been a joy running these programs because you get to see the transformation of the students from the 1st week to the 7th week. And I always tell the students that what you're doing now is very different than what I did when I was in high school. And the talks that you're going to see are probably much better than the ones I gave as a PhD student. So it's always been very impressive. And I always tell folks too, and I think Andy's heard this from me before, that if I were to apply for undergrad or graduate school again now, I probably wouldn't get it. So everyone here did a really fantastic job, and just a few kind of points for the talks today for the presenters. As Andy said, you've got 10 minutes and some Q&A, and what we'll do is, so you're not, watching the clock over here, it's around 10 minutes. Andy or I will stand up, and then if you get a little bit closer to 15, we'll start waving at you. And if you get to 15, I don't know, maybe someone will come out and pull you off the stage. All right. So with that, why don't we get started? So I'd like to bring up Brandon up here and Dr. Florentina.
Good morning. So it's my pleasure to introduce Brandon Williams. He's from the 10th grade from Winchester Trust School. So it's a pleasure. It was really nice to have you in the lab. So, and I hope that we contribute in a small part for your future research. And to say, Brandon was a very shy in the beginning. And then over the time, he opened and he talked to people and then helping a lot. Was a month of, I hope, a lot of back and forth learning from your side and also from my side and from my lab members as well. So thank you so much for all the work. And thank you.
Hello, my name is Brandon Williams. I am a rising senior at Winchester Thurston Upper School over in Shadyside. And this year I've had a wonderful opportunity to be back here at Hillman for my second summer, this time working the pathobiology site in the Alex lab under my mentor, Lapia Rodrigo. So quick introduction about my project. iPSCs, or induced pluripotent stem cells, are created by introducing specific genes. These are called Yamanaka factors, which are OCT4, SOX2, and T4 to adult cells in order to revert them back to an embryonic state so they can have specific properties to behave like stem cells. More specifically, they are used for modeling diseases due to their ability or pluripotency to model other cell types. However, due to the current efficiency of the reprogramming process, some IPS cell lines may not be fully reprogrammed and may not have the full characteristics in order to be used for research. Therefore, all iPSCs have to be characterized in order to verify their viability for research. So characterization is process of using various techniques in order to analyze the properties of something. It's quite similar to using quality control on a product. Specifically, I've been working with the IPS cell line IPSC167, specifically IPSC167 wild type and 167 gene edited. Wild type is a cell in its normal state, while gene edited is a cell with its genes modified. So the first method of characterization was doing something called immunofluorescent staining, which is a process used in order to detect the presence of specific proteins in iPSCs. Mainly proteins I was detecting presence for was NANOG, TRA160, and SSEA4. So in order to do immunofluorescent staining, the cells grew on well plates, and the cells were washed and blocked, and then they were incubated with the primary antibodies. And for the case of SSEA4 and TR160, I also had whole chest added to it, which whole chest is a blue staining dye, which locates the nucleus of the cells. And then the NANOG was incubated with the diluted secondary antibody donkey anti-mouse 594 and whole chest. And then the cells had PBS added to them in order to keep the cells wet. Then the picture of the cells were taken using EVO 7500. EVOS has different lights, active fluorescent groups in the secondary antibody, the whole chest, and in the case of TR160 SSEA4, the primary antibodies can react to. So here are the results. So the positive control is where we added the primary antibody, while the negative control was there was no presence of primary antibody. So the purple in the positive control is an overlap between the blue and the red, the blue being the DOPI, which from the whole chest, and the red being the TX red setting, which reacts to the fluorescent group in the secondary antibody. That's where the number 594 comes in. So as you can see, there is a presence of purple in the positive control versus the negative control. It's just blue with the red just meaning that the cells were dirty for some reason. And then for similar process happened with the gene edited, where the positive control had this purple overlap between the DOPI and the TX red versus the negative control, where it did not have any purple overlap and therefore did not have any NANOG present. The TRA160, the blue is still corresponding to the DOPI, while the green is the GFP, which is part of the 488 of the TRA160. As you can see, there is a blue and green overlap. And then the SSEA4, instead of being TX red, it's still red. It's RFP, which corresponds to the 555 in the SSEA4. As you can see, there is red dots that are corresponding, which are underneath the DOPI. And a similar result occurred with the gene edited, where there was GFP present in the TR160 and SSE4 had RFP present. The second method of characterization was doing an RT-qPCR or reverse transcription quantitative polymerase chain reaction, which a qPCR is a process where regions of DNA code target sequences are amplified and quantified. So in order to do the RT-qPCR, the IPS167 wild-type gene edited cells were collected and their RNA was extracted and purified. The RNA was used to make complementary DNA using a thermocycler. Then the analysis of the cDNA was done using qPCR machine with control iPSC being the reference sample. Control iPSC is collected from cells that have already been characterized in prior experiments. And the specific targets were Lin28, OCT3/4, MYC, NANOG, and SOX2, with the internal control being ACTB. ACTB is commonly used to compare expression levels due to its stable expression across cell types conditions. Also make sure there is no biological errors with the RT-qPCR. So here are the results of the qPCR. The mRNA relative expression basically means the fold change or the expression of RNA in reference to the reference sample, which is why the control iPSC is around a value of 1, which makes sense. The IPS167 wild type had a consistent fold change under each target, even though it had slightly higher values, but still overall consistent. Then the IPS167 gene edited cells had less of an overall consistent fold change due to them having at least one major outlier or one value being above or below the others duplicates. But it still overall had enough consistency for the gene edited cells to overall have a consistent fold change. So basically, inclusion, immunofluorescence staining showed the presence of the NOG, TRA116, SSE4, and IPS167 wild type in gene edited cells. The qPCR analysis showed the overall consistency of fold change values in these cells as well, which overall confirms that IPS167 cells had most of the characterization necessary so far in order to be used in research. In the future, collected IPS167 wild-type gene edited cells have their DNA sequence using genotyping to fully characterize the cells and make sure they are fully able to be used for research. The techniques used for characterization can also be used in order to characterize other iPS cell lines in the future. In addition, now that the wild-type and GI cells have been characterized with RT-qPCR, they can also be used as reference samples for future RT-qPCRs. I'd like to take a moment to thank my lab mentor and PI, Rodrigo, and the members of my lab, Ziping, Lanuza, Bo, Soon, Zhang Hao, Sean, Larry, and Alex, the directors of the Hillman Academy, Steve and Dr. Boone, and the site heads of the Pathobiology site this year, Dr. Duncan, Dr. Gao. Thank you. Are there any questions?
Brandon, excellent talk. On the outlier that you saw with the RT-PCR,
Uh,
go back a couple slides.
yeah.
Is that all from the same sample? Or is that different sample?
Those are different samples each time.
Yeah, this, you don't mind what does the gene edited mean?
The gene edited basically means that the cell had its genes edited or modified in some way that is different that's different than the wild type.
Do you know what the editing was? Like, was it to try to make it more pluripotent?
The edited, my lab mainly focuses on researching liver disease, so the gene edited was specifically so that the cell would be able to differentiate into liver cells.
An easy question for you. Which technique did you like to do better, the RT-PCR?
Probably the staining. The RT-qPCR, the process is a little bit longer and can be kind of nerve-wracking due to the fact that the qPCR is very sensitive. So any small mistake could cause like the extreme outliers in the case of the gene edited.
I was gonna ask, it was a great presentation. I was gonna ask, so how did you pick the positive and negative controls, and what did you, I know you showed the pictures of them, and how did you choose those to make sure that you could interpret your results?
So what I did, I took 5 pictures of each cell, and then I picked the one that was overall most consistent looking and also had the most had the best like visual looking so it can be easily interpreted and visualized.
Did you see when you were doing this staining, were there clusters of cells like right there, you know, where say they would be positive for an anog and your other markers? Were there other clusters within the same dish or the same well that were negative? Did you see variability amongst each one there?
No, not really. It was overall consistent in the well plate. So the laser is working.
Okay, good morning, everyone. So I'm Christy Kolarsik, and I had the pleasure of having Elise Dressman in my lab for the summer. It really was a pleasure to have her in the lab. She was really hardworking, very focused. She expected a lot of herself, and she was very driven throughout this summer experience. Even from that very first Friday presentation, which you as her colleagues got to see, she was very driven, making sure that she had everything included. And so it was really, really such a pleasure to have her in the lab, especially this is my first time being a mentor for the program. And so I would also just just add that as a part of our team. It was really a pleasure to have her. This was the most people we've had in my lab, and our space is somewhat constrained. So we did some logistical balancing of that space, but Elise was always up for it and interacted with everyone on our team so well. And so what she's going to share with you today, I won't steal her thunder, I promise. But what she's going to share too today is some work that she's done. She definitely dabbled in a lot of the things that we're doing, a lot of the different techniques to get an overview of our lab. But she really focused a lot on incorporating this new software into our lab that helps with taking our images and reconstructing the brains that we analyze to look for indicators of ALS and loss and disease. So with that, Elise.
Thank you, Dr. Kolarczyk. So I'm Elise Dressman. I am a rising junior at Fox Chapel Area High School, and I was working with ALS. My lab is working with ALS, which is also known as Lou Gehrig's disease. This disease causes physical disability, speech difficulties, and respiratory failure. This disease is usually fatal from 3 to 5 years after diagnosis. Around 10% of ALS cases are genetically inherited, but the other 90% is still unknown. In our lab, we were using Profilin-1 mouse models to simulate ALS. We then used a rabies virus to visualize axon activity. We used the rabies virus due to it moving exclusively in retrograde direction, so that's from the muscles to the spinal cord, to the brainstem, to the cerebral cortex, and the other cortexes. And then we also used it due to it moving transsynaptically, so from the dendrites up to the synapses throughout the neurons. We hypothesized structural changes in the neural circuitry occur before symptom onset in mice with ALS, and that just means that you will see symptoms cellularly before you'll see them physically. For my project, I worked with NeuroInfo, which is a brain navigation system that aligns experimental data to the reference atlas data. That is the last step of the project for the lab. In the lab, we used 2 types of mice, the control mice and the Profilin-1 mutated mice. The mice were sacrificed at different times and then were injected with the rabies virus. We injected into the TA, which is the tibialis anterior, and the soleus. Because of the different types of fibers in each muscle, we injected into the 2 muscles. The TA is predominantly fast twitch, while the soleus is predominantly slow twitch. The difference between the 2 muscles gives us insight on how the different fibers and neurons are affected. Some brains are sent to our collaborator, Diego, who is in Columbus, Ohio. He performed an MRI scan to visualize the connections in the brain. This image shows a finished MRI scan, and you can see through the colors the different types of pathways in the brain. We then take the mouse brains and we bed them into paraffin, which is also candle wax. We then use the microtome, which is this machine down here, to section the brain. The microtome is a specialized precision cutting instrument, which we use to cut the brains into sections that are 10 microns thick. That is considerably smaller than a human hair. The human hair is about 50 microns thick. We then stain the tissue with IHC, which is immunohistochemistry. Brandon actually talked about that already. This is what it looks like. And then we also stained with immunofluorescence, which is this one, which is also IF. Using the staining, we can see where the rabies has traveled and see what neurons were affected by the virus. Using NeuroInfo, scientists can see what changes happen throughout the brain. The program also allows for a quick and accurate count of different types of cells within each section. This is very useful because counting cells manually is very time-consuming. The program also allows us to create a 3D model of your brain using the Brain Atlas as a reference. So this kind of shows what you can do. And this shows just what the program looks like once you get it working. NeuroInfo takes a lot of steps setting up, and then it also takes a lot of steps setting up for imaging. First, you want to upload the images you want analyzed. As you can see here, this is all the images. So these pictures are for a process of what I see on my part. So these are all the images that I have set up, and this is an example of an image. Then you have to collapse them into an image using the serial section assembler. This is a view of what the serial section assembler looks like, and then you outline your ROIs, your region of interests. So you would either manually outline this by clicking, or you can use the program to automatically detect your ROI. Then you have to register around 3 sections into the atlas manually. This is an image of putting them into the atlas. You pick one that's far rostrally, one that's far caudally, and then one that is centered in the brain. You use those as a reference to create a transform that the software will follow for the brain. The transform is similar to an outline or example of what the brain should look like. And then, depending on what you wish to do, you are able to create either a 3D image or you're able to image them and have a cell count. These images show highlighted ROIs and then also just a picture of the brain. The brown is an example of immunohistochemistry staining, and the brown is showing the rabies. So here, 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 here. And then while in the lab, we ran into many issues. When we were cutting with the microtome, the brains would crack. As you can see here, there's a lot of cracking and shredding. And then we tried to use the cryostat. The cryostat is used, this is the cryostat right here. The cryostat is a specialized instrument used to maintain extremely low temperatures while cutting. Using the cryostat, we also ran into difficulties. The tissue would crack, shown up here and like just around this area in general, and it would also have a lot of bubbles. The picture above doesn't have a lot of bubbles, but there was a lot of bubbles. After testing both, we found out that the microtome was preferable due to better tissue integrity, and it also worked with the neuroinfo much better than the cryostat. However, the microtome was much slower than the cryostat, and obviously speed matters a lot when you're doing science. We also needed to test a lot of different antibodies, such as SST, which is somatostatin, which is this red down here, GFAP, which highlights the astrocytes, which is up here, and then DAPI, which highlights the cell bodies, which is in blue. Outside of the lab, I helped my mentor Brianna and a collaborator, Ernesto, from CMU with a preclinical study. In this study, we tested the maximum voluntary contraction of both control and ALS patients. We used an EMG, an electromyography, which measures muscle activity. This is what the EMG looks like. You have the sensors, and then you have what connects up to the computer. Then we tested several muscles, the biceps, the triceps, the TA, and the soleus. We tested using elbow flexion, which is pushing up onto the reader, elbow extension, which is pushing down onto the reader, dorsiflexion, which is pushing your foot up onto the reader, and then plantar flexion, which is pushing down onto the reader. The results show that the patients with ALS were weaker, which are shown on this chart. The ALS patients are in orange, while the healthy patients are in blue. The strength is on the y-axis, while the exercise is shown on the x-axis. ALS patients also had less endurance, however, this data is not shown. Since the lab is still continuing the histology and continuing imaging slides, and analyzing using neuroinfo, I'm not going to be there for that. They will also continue using the microtome dissection. The results from the tests will give the lab a better foundation to understanding what to focus on in the future and a better understanding of ALS in general. The lab will continue with these tests in the future and will continue using mouse models. This will hopefully give us insight to how the disease is caused in humans and how to treat it, and then also potential therapies. I would like to acknowledge my lab, Christy, Brianna, Steph, Maddie Wolf, and Paul. And then I would also like to thank Andy and Dave for Hellman Site, and then also Ernesto, Nathan, and Diego for work outside of lab. Any questions?
So I'm gonna do similar, great job, it was, I'm gonna do similar to did with Brandon. So if you go back to this slide, you have some outliers in the health controls. Do you know if those were the first measurements for those controls and they got tired after that, or is that just outliers?
Are you asking about like these ones up here?
Right.
So these ones, so this guy was just very strong, is what Ernesto told me. But this does not show, like, this shows, no. Mhm.
But that was the same patient that was strong all the way to top.
Yes, that guy was just very strong. Anyone else?
So Elise,
first question,
Mhm.
for the neuroinfo, what do you feel like the biggest challenge was and the most rewarding thing that you did?
So definitely the biggest challenge was not having the computer crash on me because our computer's not very powerful, I guess. And every time you'd upload more than 5 images, it would crash. And then the most rewarding was when I was on a video call with Nate to figure out how it worked. And then once I was done with the video call, I tested it on the computer and it worked. And I could finally get an accurate cell count, which is the best because I could not get that before. It would say it had like 100 cells. I'm like, not sure about that one. Anyone else? Dave?
Yeah, why fulfill it in ALS?
So that's a really good question. I'll get back to you later, but I'm 90% sure it's because it involves the building of the cytoskeleton, which involves the muscles. And since the muscles deteriorate, there has to be a difference in the profilin, which
You had a really unique project in that you had, both sort of wet bench work, but then you also got to interact with patients. And so for all the Hillman Academy students I've worked with over about 10 years, I don't think anybody's ever worked with patients.
really?
Yeah, that I've dealt with. So what do you like better? You like,
I liked patience. Patience as in talking to somebody. I liked wet lab because I definitely learned more and I felt more involved and I was definitely interested in it more. But the patients were fun because I got to talk to people and just kind of have small talk with different people every day.
and did that give you a different sort of appreciation about, when you see the mice and you're looking at these brains, the muscle and all that, like to see how that really relates to a person, a patient?
That was interesting because it's weird to see the translation from working with mice, small creatures, and then to humans, which are, bigger than me probably. So that was pretty cool. And it was definitely an experience to see somebody with ALS because I met one patient that survived like 15 going on to 16 years with ALS, which was kind of crazy because the fatalities rates are 3 to 5 years. Yes.
Did you learn any tips or tricks to use the microtones to make the brain not fracture like the example you gave?
No, because since our tissue is, I don't know what was wrong. We don't know what was wrong with the tissue. We would have to kind of baby it. We would have to hold a paintbrush onto the tissue and slowly crank it and then hold it and put it into the hotbed. And that was time consuming because usually the microtomy you just kind of put it into the hotbed, but we had to baby each one and it would take forever.
I'm gonna throw somebody under the bus. I think Ann Orr is over there. Yeah, she is an expert at doing tissue sections like that. Okay.
Yeah, we were in
So
there.
it's your fault.
No, it's definitely not your fault.
I would not say it's your fault. No, we got to use her cryostat, which was amazing.
So why do you think the brain is so hard to section compared to others?
We don't know. Maybe we were testing each one and we're trying to see either if they were sitting too long, if they were sitting in the wrong solution, or it's just an experience in cutting. Because obviously I'm not a pro at cutting with a microtome. It's the first time ever using one this year. Anyone else?
Najee, you're next in CD.
After treatment for throat cancer from smoking, with home hospital stays and surgeries, you realize the true cost is a lot more. You can quit. For free help, call 1-800-QUIT now.
Hello, good morning all. I'm Siddhi and today I'm here introducing Nachi, who worked with Dr. Bhushan in like acetaminophen-induced liver injury model. He was involved in doing like downstream experiments for EGFR, use of EGFR inhibitors in hepatotoxicity model, where he tried to look into injury and like other like DNA damage assay, and he was involved in doing immunoblotting analysis. And I really say he was like very a quick learner, and it was also helpful for us to have him. Like while I was writing papers, he was also helping with my experiments which were ongoing. So really happy to have him in the lab. Thank you.
Hi everybody. My name is Naji Hader, and I am a rising senior at Winchester Thurston School. Just here, maybe like 5 minutes away. This summer, I was blessed to have the opportunity to be a part of the UPMC Hillman Cancer Center Academy, in the pathobiology site. My lab, I was under Dr. Bushin, and my mentor was Siddhi. This summer, I worked with the project of EGFR inhibition and how it reduces liver injury following an acetaminophen overdose. So starting with some background, what is acetaminophen or APAP? Acetaminophen is a widely used over-the-counter pain reliever. For example, it's in Tylenol, and it's in over 500 other FDA-approved over-the-counter medications available here in the US. At safe doses, APAP is very effective and completely non-toxic. However, high doses can cause a severe liver injury. So looking at what happens when you take a high dose of APAP and how you can overdose on it. APAP is metabolized in the liver, and the one toxic metabolite of APAP is called NAPQI. Normally, at a safe dose, NAPQI is it's completely safe because glutathione or GSH binds to this NAPQI and allows it to be safely excreted through urine. However, in the liver, humans, we only have a limited supply of glutathione. And when this GSH glutathione runs out, the NAPQI causes downstream DNA damage. And in this severe overdose, the DNA damage caused by NAPQI, not directly caused, but like a domino effect, this DNA damage, it's too significant for the liver to regenerate. And this ultimately causes acute liver failure, which often results in death. The only current treatment for an APAP overdose is NAC. NAC, it kind of acts as like a substitute for glutathione, but NAC only works in the first 8 hours after an acetaminophen overdose, and majority of patients don't come in soon enough to be administered NAC, so this creates a need for a new solution that can help patients that come in after this early time frame. And then kind of zeroing in on my project and what I did, I worked with EGFR or epidermal growth factor receptor. And EGFR, it typically aids in cell growth and liver regeneration. For example, after a partial hepatectomy, which is when up to 2/3 of the liver is cut, EGFR, it supports healing and is really beneficial in allowing the liver to use its regeneration to its full capacity. But its role in acetaminophen injury is very different. Prior studies, including some of ours, show that blocking or deleting EGFR actually lessens liver damage from an acetaminophen overdose. And that EGFR may worsen injury in an overdose, and this is by boosting stress signal. So with all this, we hypothesized that the clinically approved EGFR inhibitors, afatinib and osimertinib, will reduce liver injury by decreasing DNA damage following an acetaminophen overdose in mice. Afatinib and osimertinib are both already FDA-approved drugs. Right now, they're used to treat certain types of lung cancers, and they work the same way in lung cancer where they inhibit this EGFR. Right now, I think osimertinib is more commonly used as it's more effective for treating lung cancer, but both are widely used, and we were looking at how they impact liver injury after an APAP overdose. So for our methods, we used male C57BL6 mice, and they were given a toxic dose of acetaminophen, which is 500 milligrams per kg. And 1 hour after they were given this toxic dose, they were treated with either a saline, which is like a vehicle, it's a control group, afatinib or osimertinib, all at 20 mg per kg. And then we assessed the liver damage 6 hours later using serum ALT, H&E staining, and tunnel staining. So first, looking at the serum ALT, what ALT is, it's a protein that is released when cell death occurs in the liver, and it's released into the bloodstream. So the higher the ALT level, the more injury there is in the liver, the more cell death has happened. So starting by looking at the A500 plus vehicle group, which is the control group we talked about, that has the highest level of ALT, which signifies the highest amount of liver damage. That's around 3,700. That's followed by the A500 plus afatinib group. Afatinib is that first EGFR inhibitor. And while there's a small decrease, it's non-significant. And then lastly, looking at the A500 plus osimertinib group, we see a massive decline. That's at around like 1,400 as compared to 3,700 in the control vehicle group, which shows a significant decrease and significantly less liver damage. The next piece of data is looking at H&E staining. What H&E staining does is it stains the live nuclei a dark purple and cytoplasm, dead cells, and other things present a lighter pink. So once again, looking at that first group, A500 in the vehicle group, we can see while there is a little bit of dark purple, majority of this tissue is very light pink, dead cells, almost no nuclei in these dead zones over here. That shows a lot of cell death, liver necrosis. Next, looking at this A500 plus afatinib group, we see a slight decrease, but there's still this massive dead zone and significant liver necrosis. And then lastly, looking at A500 and osimertinib, we see a significant decrease in cell death and liver necrosis. Even in these dead zones, there's still tons of live nuclei, and overall, the liver architecture is much better than in these other two samples. And then the last piece of data we're going to look at is tunnel staining. What tunnel staining does is it stains cells with damaged DNA or fragmented DNA a dark brown, and live area is this kind of gray blue. So once again, starting at the A500 vehicle, the control group, we're going to see that there's damage all over, very dark brown, signifying a ton of DNA damage and cell death. Moving on to afatinib, there's still a lot of damage present, but it's not as dark and not as widespread as in this sample. So that shows slightly less DNA damage, but still very present. And then lastly, looking at the A500 plus osimertinib group, we can see by far the least damage, and the brown is much lighter, showing the least amount of DNA damage overall.
If this is it, he's not listed.
And then looking at a data summary,
We got.
the mice treated with afatinib, osimertinib after an APAP overdose overall showed lower serum ALT levels. That was that graph indicating less liver damage. H&E staining, the pink tissues showed reduced necrosis and better overall liver architecture compared to the vehicle. Tunnel staining, the brown, the last one we looked at, revealed fewer dying cells, indicating reduced DNA damage. And overall, osimertinib did have a stronger protective effect than afatinib across all data. So conclusions, what can we actually do with this? What does this actually tell us? EGFR inhibition after an acetaminophen overdose appears to protect liver cells from damage and death. Both inhibitors reduce key markers of injury, ALT, necrosis, and DNA fragmentation. And just looking into the future, what can this actually do? These findings support this idea that EGFR inhibitors could be repurposed as emergency treatments for an acetaminophen-induced liver injury, especially when patients present too late for that only current solution we talked about, which is NAC. And then thank you. My acknowledgements. go out to the Bushin lab where I worked. Siddhi, Dr. Bushin, Jillian, and Rome, they all, those are the people I worked with every day. They were great. They taught me so much and helped me grow from what I was two months ago to what I am now. Also, everyone else, the Michaelopoulos lab, the whole Department of Pathology here at Pitt, and then also the Hillman Academy, Dr. Duncan, Dr. Gao, thank you so much for running this site the way you did. It was great. And then the people that run this high school's Cancer Center Academy, David Boone, Joe Ayub, and Stephen Jones. Thank you.
So really, really nice talk. So you had a really nice graph, with statistics like showing us like the serum ALT levels. For your, you have those two other histology-based metrics. Do you have, ways or ideas about how you could quantify those and then show us, basically demonstrate your.
Oh, yeah, I
what
understand.
you're seeing is trend?
So obviously my time is over, but I think soon we have a software that actually like can go in and like measure what cells are alive at what rate, like where, because obviously these are hepatocytes, they're like different zones where you'll see like more dead cells or more live cells, and where like dead cells affect more in liver function. We do have software that like counts living cells, dying cells, where, how, and can quantify them in a graph. I'm just leaving now, so thank you.
Yeah, I don't know. ASFT. So you show that the reveals after the treatment with the drugs. Or does not values like close to normal or?
You're talking about this graph? Are these values close to normal? Yeah.
How close the treatment goes to a non-treatment?
Are you're asking a normal APAP overdose patient or a completely normal?
No.
Like a completely normal person, no overdose. A completely normal person, no overdose is gonna be like not even like 100. It's gonna be like 30. So very low. ALT is only a marker of liver injury, so it won't show up unless we see liver damage. Yeah.
Yeah, so you mentioned that acetaminophen was more efficient, especially with treating lung cancer earlier.
Yeah.
And obviously your results have shown that acetaminophen
Yeah.
is better at
Yeah.
stopping liver damage. Is there any reason for that? Is it just a better EGFR inhibitor than
Yeah, I got you. So I actually don't know anything about really like what actually the difference between osimertinib and afatinib is. That's definitely something that we could look into in the future, understanding why. That's a great question. I just don't know.
So you said the main issue with like the current treatment is that it can only be administered like within a certain time frame. What is different about like these two, like the triple dosing that makes it so they can be administered after the other one?
I got you. Okay. So you're talking about NAC. So what NAC does, as I said, it's like a substitute for glutathione GSH. And what it does is it takes that NAPQI and it allows it to be safely excreted. But by the time 8 hours comes around after an overdose, NAPQI has already like started that chain reaction, because NAPQI doesn't directly lead to DNA damage and cell death. It's like a domino effect. NAPQI goes into the mitochondria, releases AIF, and then AIF goes into the nucleus, and then you know what I'm saying? So for this EGFR inhibitor, EGFR, it can work in those later stages when that downstream damage has already begun.
Good job. Why only male mice?
So certain hormones in female mice kind of make them comparing female mice to a human model doesn't correlate anywhere as well as just looking at male, just certain hormones. And it's easier to line up. This is like I think it encompasses a lot of mice studies where looking at female mice and their hormones just kind of like throws certain certain like ways that mice a mouse's body reacts like a lot different from what a human body would react.
And I had one other follow-up question.
Yeah.
Based on the question with this graph earlier.
Yeah.
Do you have a hypothesis what levels, if we use, Alice's, the marker, do you have a hypothesis of how low that needs to go in order to not be so damaging that it causes death in?
Yeah, okay, I understand. So that's definitely something we're gonna look into later. But I think I mentioned it earlier, but partial, actually, let me pull up the slide. Okay, so this one, after a partial hepatidectomy, the EGFR can support healing. And that's at that 2/3 stage. So a liver can regenerate itself. It has a very strong regenerative capacity, but only if, obviously, if like 95% of the liver is gone, it can't regenerate. But up until 2/3, it can still regenerate. So we just need to get it down to that point. For this study specifically, we were just looking at whether EGFR can even work. It was more of like a proof of principle, like because if EGFR doesn't even work, then there's no point in looking further, seeing how it can actually impact the human body, seeing how it can actually work. Like for example, another issue that was that we saw in this study was that we're injecting the mice with these EGFR inhibitors 1 hour after that APAP overdose. At that 1 hour, you just give them NAC because NAC is completely, it works every time. So this is more of a proof of principle, just understanding how EGFR actually interacts. EGFR interacts with these EGFR inhibitors and see how that impacts the overall injury and damage. But that's definitely something I think that we are already looking into and seeing how it can actually impact real patients.
You know, if like 1 hour afterwards, you said nap up to 8 hours, but if it's 1 hour afterwards, just use glutathione instead, or was giving glutathione not work because it's intercellular?
I got you. Yeah, glutathione it takes a really long time for it to go through and actually be able to be used in the liver. It just takes a while to metabolize and actually go in.
Okay, aren't these guys amazing? Like, those are three outstanding talks. We're perfectly on time. It's exactly 10:30, so we're gonna do a 15-minute break. And we'll resume at 10:45. If anybody needs the restroom, it's right across the big lobby over that way. So, and during this break, ask the three speakers hard questions, please. I assume we'll have a few people trickling in as we get going here, but next up, we're gonna invite Ezra and Dr. Perkins to come do the introduction. What? Or with her, but. I didn't see anybody out there. I don't know if they went up to the labs. They want to manage.
I don't have any good jokes.
I wasn't started.
Sure. All right. Good morning, everyone. Thank you for being here. It's my pleasure to introduce Ezra Hardy, who's been working in the lab with us this summer. Ezra really has been great to have in the lab. He's really picked up on all the techniques we've used very, very quickly. I know he's going to show you some of them today, but I really kind of threw him into things to get him some exposure to all of our major fundamental methods we use in biomedical science. Something that really stuck out to me with Ezra is the questions he would ask. These are very intelligent questions, which really relate to me that he was understanding everything that we were talking about, which are very complex things. That may reiterate what Dave was saying about some of these students and their presentations is these are more intelligent questions than I was getting from graduate students in recent years. So very bright. He also may have helped us optimize our methods for preparation of tissue for RNA extraction. So I appreciate that. And I'm sure he will be very successful in whatever discipline he chooses, but I certainly hope it's biomedical science because we could use more scientists like I think he would be. So thank you, Ezra.
Thank you.
Okay. Hello, everyone. As mentioned, my name is Ezra, and this is my project, Understanding the Role RAGE Plays in Obstructive Lung Disease. Now, what does that all mean? Well, let's start with obstructive lung disease. Obstructive lung disease is not one single disease, but rather a group of diseases that all affect the upper airway. They can cause mucus hypersecretion and inflammation. If you look up at the photo on the A side, that's a normal airway. You can see nice and clear. But on the B side, that's the airway of someone with asthma. You can see there's a lot of very clogged, and that can make it a lot harder for someone to breathe. Now there are different types of inflammation, but the main two that are important for this presentation is type 1 and type 2, and I'll get into a bit soon what that exactly means, but the mechanisms behind both types of inflammation is not currently super well understood, which can make it hard to find an effective treatment for these diseases. So what is RAGE? RAGE is short for the receptor for advanced glycation end products and is a cell surface receptor that is found all throughout the body, but is mainly produced in the lungs. Now how it works is basically if you see this little diagram, there can be like irritants or allergens that enter the lungs, and RAGE will detect that, and it'll release IL-33, and then depending on certain factors, it can either result in type 2 or eosinophilic inflammation or type 1 neutrophilic inflammation, the blue one. So my mentor previously did a study on how RAGE mediates type 2 inflammation. So one of the questions we had was, how does it mediate type 1 or neutrophilic inflammation in mice? So to do that, we had a mouse model, and basically we had 10 wild type mice, which are just regular mice, and 10 mice with the RAGE gene knocked down, meaning they didn't produce any RAGE. And then they were exposed to either saline or a combination of cyclic diGMP and IL-33. Now, the reason we had that combo specifically is if you look at the bottom, like 4 square, you can see that saline and CDG on their own both didn't produce any inflammation, while IL-33 on its own produced specifically eosinophilic inflammation. But if you combine CDG and IL-33, you get neutrophilic inflammation. So because that's our main sort of target, that's why we use that specifically. So we exposed them every day for 4 days, and at the end we collected the bronchoalveolar lavage fluid, or BALF for short. Basically, how we did that is my mentor would inject fluid into the mouse's lungs, and then he draw it out, and then any particles that were in the lungs would now be in the fluid. So using this lavage fluid, we examined the lung cells and measured the cytokines by an ELISA. Now, an ELISA is a process where basically it starts like at a 96 well plate, then we add a capture antibody to each well. So you can see in the bottom, the capture antibody would basically stick to the bottom of the well. And then we'd wash in between each step, and then basically we would add our specimens and our target molecule that would bind to the capture antibody. Then we add the bitenolated detection antibody and HRP, which would attach, then we added the substrate, which would cause a reaction if HRP was present, and that would cause a change in color. So you can see in the top there, some of the wells are more darker yellow, while others are like more clear. And then basically using that, we put it in a spectroplate reader and measured the absorbance, which we can use to estimate how much of our target molecule is present in our specimen. So it's like a little chain. So another thing we did with the BALF is we put it on some slides and we stained it. So this is a nice visual, so you can see that the saline in both cases didn't really produce any inflammation because that's our control. And in a wild type with the IL-33 and CDG, you can see there's a lot of cytokines, and you can see that that's evidence of pretty severe asthma, while with the RAGE knockdown, you can see there's a lot less. There's still some inflammation, but it's not as noticeable as the wild type. So getting back to the ELISA results, this is what we collected. So the black is the wild type, while the blue is the rage knockdown, and you can see that the rage knockdown is very similar with the saline and the IL-33 CDG. The main one that's kind of the outlier is the IL-33 and CDG plus the wild type, which you can see has a lot higher levels than all the others. Now moving on, we have our next experiment. Now this experiment was mainly focused on how eosinophilic inflammation in human cells. So basically we had these cells that were collected from people usually who are getting lung transplants, and they would donate some cells, and it was either from people with no lung disease or people with chronic obstructive pulmonary disease or COPD. And basically these cells are put in a well with either a RAGE inhibitor, which is that AZR, or a control, which was had RAGE, no RAGE inhibition. And these cells were stimulated every 2 days for a week, at which point we collected the media and we measured the chemokines using another ELISA. So that's the results from this. That's the top two graphs. You can see the vehicle is the black while the RAGE inhibitor is the blue. And you can see that there's about a 30% difference showing that the one with no RAGE inhibition was like more receptive to inflammation. Now, another thing we discovered is if we looked at the difference between non-diseased and COPD cells, so the COPD, if this is the bottom, the COPD cells are blue while the non-diseased are black. And you can see that the COPD had a much higher reception to inflammation, which is just something interesting I thought I'd share as well. So what does this all mean? Well, because the results of our mouse experiments corroborate the results of previous mouse experiments performed by our lab, we can conclude that RAGE is also necessary for type 1 neutrophilic inflammation, and that pharmacological inhibition of RAGE will reduce the inflammatory effects of IL-13. Now, some future directions we could pursue is repeating the mouse model in IL-33 receptor knockout mice. We could also repeat the COPD experiments with higher doses of RAGE inhibitor as well as a genetic knockdown of RAGE, which my mentor thinks might make the results even more concrete. And future experiments would just generally be investigating the mechanisms by which RAGE mediates IL-33 and IL-13 signaling. Now, my acknowledgements are pretty sparse. I'm not sure what happened, but I had like a more finished version, but anyway, I would like to thank, even though it's not written here, I would like to thank my mentor, Dr. Perkins. I would like to thank the people at Hillman, including Stephen Jones and David Boone, and I would like to thank the site heads at Pathobiology, Dr. Duncan and Dr. Gal. And I would like to thank, yeah, that's everybody. Thank you. Yes.
Yeah, so I mean, obviously based on your experience, less rage seems to be better, right, in decreasing inflammation. And so my question, and this might be a little beyond scope, but are there any other known like uses of rage, either in mice or in humans, that like decreasing it could be negative in some way?
Well, it is found, like I said, it's found all throughout the body. It is mainly in the lungs. Now, there was a previous experiment, um, I meant, that my mentor mentioned, where they looked at how the RAGE inhibitor affected Alzheimer's. Now, that didn't end up leading to too much. But we're thinking that maybe this RAGE inhibitor could be repurposed for disease. Yes.
Been talk to repeating the first experiment you did with the RAGE knockout mice, and instead just use wild type mice with pharmacological inhibition of RAGE with the exposure to the irritants we do.
That's a good idea. I mean, um, I'm not sure that, I mean, that could definitely be something to pursue in future directions. I'm not sure what the difference would be, so I mean, that's obviously something that we would test, but if we did that, yeah, that's a good idea.
It looks like there's one question on Zoom.
Oh, good question.
Hi, Ezra. It's Dr. K. How are you?
Oh, hello. Hi, Dr.
Good.
K.
Good. Nice job on your presentation. Thank you.
Yeah.
Interesting topic. I had a question. Are there any RAGE inhibitors used in the clinic right now to treat any of these inflammatory lung diseases?
Well, there was, like I said before, with the Alzheimer's, there was a human trial with that, which is good because it means that it was approved for use in human testing. I don't know currently if there's anything that's actually being used like in a clinic for treatments of something. Yeah, but it was something that was tested by other researchers.
Gotcha. Gotcha. What was kind of your favorite part of this whole experience so far this summer? I know you've done several different, lab environments.
That's hard to say. I mean, I think it's well, I mean, it's just been nice to sort of be in a different environment, just tackle like a different topic. I enjoyed this year. Like I'm not sure specifically what I would point to as like my favorite part. I just enjoyed the whole of it together. I like that my like results felt more concrete this time.
Excellent.
Yeah.
Okay. Didn't mean to put you on the spot, but sounds good. Very nice job.
Good questions.
All right, I think we'll move on.
Okay.
So next up, we have Alberto and his mentor, Dr. Wendy Mars.
Good morning, everybody. I would like to introduce Alberto. He is a rising junior at Alderdice and has been a delight to have in the lab. Alberto learned that I'm often not in the lab, but he had a huge support system of everybody else. And I have to say, when you sit and talk with Alberto, he makes you feel good because he has wonder. He still has that wonder. And he was a lot of fun to be with. My favorite line, which he uses a lot, is, "Oh, that makes sense." And it was really fun to work with him. I think up until yesterday, everything didn't really come together, but I think once it came together, he really is on to something, and probably we're going to continue along these lines because he really found something interesting. All right.
Thank you. Okay. So first, I would like to thank everyone for being here. One of the things about Hillman is doing research, but a part, Hillman, that I also like is being able to share your findings and share the research that you did. So as to who I am, I'm Alberto Gildingers. I'm a rising junior at Taylor Alderdice, and today I'll be giving a presentation regarding a preliminary study on human alpha defensins in an aberrant role in liver disease. So my presentation will go as follows. I'll start with an introduction and backgrounds, then move into my hypothesis, the methodology, results and discussion, and then finish with the conclusion and acknowledgements. Oh, it worked. There was a corrupted file, so I didn't expect this to work. The first step to understanding this presentation and broader research on alpha defensins is understanding what alpha defensins are. And the answer to this is that they're innate immune response peptides. More specifically,
they're antimicrobials, and they have many functions throughout the body, but one of their main functions is pathogen regulation. And they perform this through membrane disruption, which they bind to the membranes of pathogens and poke holes in the membranes, ultimately causing the pathogen to die. And this role as a pathogen regulator is very important for broader hepatocellular functioning because it helps maintain homeostasis. A buildup of pathogens and bacteria in the liver can cause malfunction. It can prevent metabolism. So it's very important that alpha defensins come in and make sure that there isn't this buildup. Now, as for some more things about alpha defensins, they differ, or DFAs 1 through 3 differ only on the basis of one amino acid. And while this seems like a very small difference, and is a small difference, it causes vastly different functions. And another difference is that you can have between 2 and 20 alleles of A1, but you can have between 0 and 2 alleles of A3. And in fact, A3 can be inherited as null. Now, there is a relationship between the quantity of genes, A1 and A3, and the likelihood for developing cirrhosis. And this didn't change on the basis of type of liver disease, ALD, which is alcoholic liver disease, or MASH, which is metabolic dysfunction associated steatohepatitis. And further with the quantity relating to disease, unpublished data indicates that people that have ALD have a higher gene ratio of A1 to A3, and this suggests a relationship between disease severity and alpha defensin genes. But this is more on the genetic side, and what we were trying to do was apply this into a more visible and protein-related sense. And this leads to our hypothesis, which was that the number of immune cells in ALD and MASH correlates with disease severity. And this is trying to associate the relationship between the quantity of proteins, alpha defensins, and disease severity. And this builds off a point that constant inflammation leads to a higher likelihood for cirrhosis and fibrosis, which worsens over time, and has ties to things such as hepatocellular carcinoma. And alpha defensins, as I mentioned, as innate immune response peptides, play a key role in this inflammation and the immune response. Now, to test this hypothesis, the first thing we had to do was find a way to view alpha defensin expression. And we did this through immunohistochemistry. This worked via a primary and secondary antibody binding to the alpha defensin, and then this was amplified. And alpha defensin expression would be shown as brown precipitate or brown staining. And our initial data sample had 23 male samples and 19 female samples. And of these samples, we had 7 donor liver samples with chemo. And donor liver is from a tumorous liver, but a resected part. So cancer patient, but it's a normal sample that we're looking at within the liver. And then we had 6 donor liver samples that didn't have chemo treatment. We had 17 alcoholic liver disease samples. We had 9 MASH, and then CP is the Child-Pugh scale, and it's the severity of the disease, with C indicating a higher level of disease severity. And we had 13 with disease state B and 13 with disease state C. And this is kind of where everything turned in a 180, because originally we were trying to look for association between the quantity of immune cells and then disease severity. But if you look at this, the main difference isn't between the single immune cells, which are shown here by the light blue arrows, and here on this other one, but staining in the biliary duct epithelia. And staining is not supposed to be in the biliary duct epithelia, but the difference in the samples is not on the basis between the quantity of the single immune cells as much as the basis of staining in the biliary duct epithelia, which you can see here and here. And this caused us to kind of switch the study. We then changed it to the presence of staining in the biliary duct epithelia, and we graded this presence on a scale of 0 to 3, with 0 indicating minimal or no staining and 3 indicating a high degree of staining. And because the scale is a bit subjective, we tried to reduce this via independent grading and a blind analysis. And this allowed us to get some results. First, we have the non-statistically significant results. We found no difference in the degree of alpha defensin expression on the basis of sex. And we also, this goes against our original hypothesis, there was no difference in alpha defensin expression on the basis of disease severity, with C indicating higher disease severity. But as for statistically significant results, we did find a relationship between alpha defensin expression and the type of disease, specifically between cancer and ALD and between ALD and MASH. And after we saw this, we looked further within the cancer samples. We found a relationship and a very statistically significant relationship between the presence or absence of chemotherapy and alpha defensin expression. And this is the same thing with the liver resections that I mentioned previously with the donor livers. And building off this, we wanted to see if this relationship held up, the chemotherapy exposure to alpha defensin expression. To do this, we used cell culturing to isolate for sorafenib, which was our chemotherapy treatment. And we had 24 wells with 12 receiving a DMSO control and 12 receiving a sorafenib fix. We did this at 24 and 48 hours respectively. And this is what's interesting. We actually found that you could induce alpha defensin expression. This is the 24-hour sample with sorafenib, and this is the 24-hour control. And we kept everything to the best of our ability constant, and this pattern held up throughout the samples in the wells. And this is showing that the sorafenib was the thing that was causing this expression, or sorafenib can be one of the things to induce alpha defensin expression. Now, as to the discussion, we have some statistically significant relationships. One of the things that wasn't statistically significant was our original hypothesis, the relationship between alpha defensin expression and disease severity. That did not hold up, but there was a relationship between type of disease and alpha defensin expression and between the presence or absence of chemotherapy and alpha defensin expression. Now, the significance of this is a generalization to broader disease. I know in my title I said aberrant, and I kind of didn't talk about that as much, but alpha defensins are not known to be made by biliary duct epithelia, and we're seeing this, something is causing this staining. It's not supposed to be there. That's one of the things that we're looking at. Something is going wrong along the way, and we're trying to figure out what that is. And alpha defensins, they tie into lots of things. They're throughout the body. And this ties into a generalization to broader disease. In the limitations, the biggest limitation was about the samples. Because it was blind, we don't have a lot of the background about the samples themselves. We don't know what caused the patients to die, etc. And that would prevent us from generating a broader relationship because there are some variables that we just aren't sure about. The thing is, this is a preliminary study, and I want to stress that. Then the other thing is the subjective staining. The way we tried to reduce this was through independently verifying the samples. Dr. Mars and I both looked at the samples, we graded them, we noted the relationships, and those matched, I want to say, in like 39 of the 42 samples. And then actually like 2 days ago, I checked the ones that we didn't agree upon, and those did not significantly influence the relationship, the pattern that we already knew. But because there's a lot that's unknown about the samples, this isn't as much of a limitation because this is a preliminary study. This is an introduction more so. So the conclusion was that biliary duct epithelial cells produce alpha defensins under specific stressors, and this is an unknown and novel finding. And then this suggests a relationship between alpha defensin production and broader disease prognosis. And because this is a preliminary study and this relationship cannot be concluded, this suggests further studies. Then I have some acknowledgements. First to Wendy, who looked over this presentation, who explained alpha defensins to me. Actually, until like yesterday, one of the biggest things that I didn't understand was between the acuteness and alcoholic liver disease in MASH. To Anne, who taught me the staining techniques and was the person who directly oversaw me. To John, who allowed me to tag along with him. He showed me Western blot dissections. To George, who allowed me to use his microscope and who also explained the induction. To Sid, who allowed me to do dissections. And to Rodrigo, who provided the samples and made sure they were blind. And then to Hillman, David, and Andy, they helped with the Monday and Friday work in progresses. They did a lot of backgrounds. And then to the Department of Pathology for hosting me to Hillman. It was a great opportunity. And then to UPMC and University of Pittsburgh. Then there were also Wednesday sessions. There were lots of people from the School of Medicine who came over. They helped, they explained their work trajectory, lots of things such as that. Thank you. Yes.
So I love, that was a beautiful histology slide you showed with the ducks lighting up the opposite incidence. And I've been asking for that for 6 weeks. So that's beautiful. The thing that caught my eye was on the left image toward the lower corner on the right. I see a lot of hepatocyte nuclei.
The dark staining, my left.
Yeah.
This one.
Yeah. So
Okay.
down below, there are a lot of hepatocyte nuclei that are lining up to the right a little bit. Yeah, like a lot of those. What do you think about those hepatocyte nuclei lighting? What is that? I don't really see that on the right.
I'm not as sure. Originally, when I was looking at those, I thought it was like they're like spilling their guts. There's this background staining. I think that's what I was told, but I'm not exactly sure. You're talking about the brown staining.
Yeah, the nucleus right in the middle is a nice brown. Anyway.
So Andy, my way in here.
Sure.
Okay. However, when he did these, he actually wrote down everything that he observed. So I can go back in the data, and it was not unusual to see hepatocellular staining, but because it would have been a lot more subjective to look at the hepatocellular staining. So we focused on something where we got our data, which is the ducts. But yes, there is hepatocellular staining as well. And that was also a big surprise. I didn't think about the fact that it shows in this as well, but it does. You can see there's probably some.
Yeah.
Also on the right, you really.
I hope I'm in the right place.
I think there's. Yeah, one of the things is these show different like segments or parts of the liver. We like categorize like regional throughout no staining. I think there was one that didn't have that. Yep.
Albert, I know you were looking at chemo-treated liver, not the cancer portion. Did you look at any of the cancer portions with some of the immune exclusions that start happening in cancer to see what the staining looked like between the cancer area versus non-cancer area?
I don't believe so. I'm not quite sure, but I do not believe so.
We talked about, we don't have.
Yeah.
And were the chemo treated all treated with the same chemotherapies for your patients, or do you know the most of them?
No, but that's why we did the cell culturing to try and isolate, and those were sorafenib, and we kept that constant. Okay.
All right, next up, we have Kai and her mentor, Ian.
Hi guys, my name is Ian Mooney. I was Kai's mentor this summer in the Vargas lab, along with the help of our other mentor, Grant, who unfortunately couldn't be here today. And whenever Grant had to exit the lab, I'm glad he's sitting back there, was a great unofficial third mentor to Kai as well. So we're gonna kind of switch gears and talk about some cell biology now. I'm not gonna take up too much time, so I'll let Kai take it away and explain the rest. Thank you guys.
The effect of AAK1 inhibitors on AP2 and alpha-synuclein localization. Okay, so for a little bit of background, alpha-synuclein is a small but abundant protein in the central nervous system. It plays a role in synaptic vesicle endocytosis. Some people think it may also regulate the bending of membranes, but what we do know is that a mutation of this protein leads to familial Parkinson's disease. This right here is like a 3D version of alpha-synuclein. Okay, and synaptic vesicle endocytosis, basically SVE. SVE is the process in which neurons recycle synaptic vesicle membranes after neurotransmitter release. AAK1, specifically, out of all the proteins we're focusing on is AP2 and PAP2. AAK1 basically phosphorylates AP2, creating phospho-AP2, which is PAP2. PAP2 travels to the cytosol, here, let me see, travels to basically like the cytosol and binds to the membrane. Without AAK1, AP2 does not travel to the membrane and preventing SVE, because without AAK1, AP2 cannot phosphorylate. Okay, so we modeled HEC cells and used an AAK1 inhibitor, which is in 3 and 4 of both the cytosol and membrane. The question we was answering is, what is the effects of AAK1 inhibition on proteins membranes localization? Our hypothesis for this was the AAK1 inhibitor prevents AP2 and synuclein movement from the cytosol to the membrane. Okay, and this is a little bit of our methods. So we use 4 cultures of HEC 293 cells, 2 treated and 2 untreated. Our untreated were our controls and the treated were treated with the AAK1 inhibitor. The cells were, basically the cells were stripped in a membrane and cytosol were separated using ultracentrifugation. And the protein concentrations were determined by a BCA. Then we did gel electrophoresis using an SDS page gels following immunoblotting. Okay, this is basically more of an in detail part of our methods. Basically of how we stripped the gels, basically of how we stripped the cell plates and everything. So we lysed with a hypertonic buffer before passing through a 26 gauge needle 20 times. Let me tell you, that was tiring. And then we got the nuclear fraction in cell debris separated from the supernatant using the ultracentrifuge, which span at 150,000 xg. The first fraction we got was the cytosolic fraction. That's important. And then after 3 washes, we got the soluble membrane proteins. That's also important. That's the ones we need. This is our control of our systolic marker of tubulin. Basically, this is our control because tubulin is only found in the cytosol, and is basically to help us show that the process actually worked in replicating the SV process. And this is our sodium potassium ATPase. This protein is only found, this is also another control because this protein is only found in the membrane. Did you see? Okay. And we did the experiment with six other gels. The first 2 sides were just our controls. These ones are our results for the next few gels. Basically, these were the good gels, even though this one is a little off. This is our PAP2. This one's a little off. It's a little faded, but as you can see over here, both the untreated cells show a band. And that's because they weren't inhibited. Basically, the AP2 met with the AAK1 and created PAP2. The treated ones did not get the AAK1 because of the AAK1 inhibitor, so they did not become PAP2 and were not seen. This is the clathrin. Basically, these are good results because they're stronger inside the cytosol and less in the membrane. That's as to be expected. This is the tubulin again. Yeah, these were good results of that. These were these three gels. The next three gels showed little to no band, and so we had to alter them for the second time. But this is the first time around. So this is the AP2. As you can see, it's a little blurry. We did a 1 to 1,000 dilution for this one. This is the sodium potassium ATP ACE. This one also very blurry and showing up as something different. We think it's because for this one, I think it's because I'm accidentally mislabeled something. This was on me. And for the alpha-synuclein, it shows nothing. So for the second tries, we decided, this is the second tries. The second try for alpha-synuclein, we just changed the concentration and added more and added a 1 to 5,000 dilution. And as you can see, there's still nothing in the synuclein. For this one, we got the right protein and we got good results from this one. We kept the same dilution, obviously. And this one, we changed the concentration. It's slightly better results. The bands are a little more clear than the first one, but they could be better. This is the summary of results. Basically, the AAK1 inhibitor does prevent the movement of AP2 on the membrane. Suggests that the LP treatment does influence the phosphorylation of AP2. And we also think that alpha-synuclein may be undetected due to its smaller size and insufficient amount of protein added to the gel. Most target proteins in the HEC293 cells were detectable, like the tubulin, the clathrin, in sodium, potassium. Those were all detected. Moving forward, various cell lines will be transfected with the alpha-synuclein and protein concentrations will be analyzed in the membrane and systolic fractions. Oh, my acknowledgements. My PI, Karina Vargas, she's not here. My mentor, Ian Mooney, my other mentor, Grant. Oh, and Gladys Molina, I'll take her as my mentor. Yeah. And to the pathobiology lab, and I don't know these people, but they're also in the Vargas lab, so thank them. Yeah. And then to Andy and David, thanks, guys. Yeah.
So since we were studying SVE, which is a process that is really dysregulated in Parkinson's, why use a Excel model rather than like a neuronal model?
HEC cells basically replicate the SVE process with majority of the proteins that we use, especially alpha-synuclein.
I had a question. The first Western blot we've seen, can you just kind of go through kind of like how the methods work for this and what we're looking at, with those dark bands?
The first one,
Yeah.
the controls?
Yeah, how does this assay work?
The method?
Yeah.
Oh, okay. So basically, after we're done running the gels, we put them on a membrane, and then we used another like a transfer electrophoresis to transfer those onto a paper, and then we put them in the boxes and add our primers and stuff to it.
Second part of it, did you guys do any or thought about doing any immunostaining to look at the actual like we saw a bunch of immunostaining in the previous talks. Do you let's talk about doing some of that, or have you tried any of those studies this summer?
No.
I can put it, but
Yeah.
we originally were going to transfer cells with alpha-synuclein tag with like an cherry tag, and then in that first round we were gonna just have a little cover slip with the cells on it and just be able to confirm that the synuclein had got see the right transference, but unfortunately that.
Those were, your Westerns are very nice, and I like the way you explain how they're, you're optimizing things and fixing things. What was the difference from the first couple you showed, which were, you were talking about how the fractionization worked with the cytosolic fraction and the membrane? Those were absolutely beautiful. Did you do those too? Or were those sort of like samples you inherited or blocks?
Oh, we did those too.
Yeah. So what changed from the first couple that were like, they look like you'd see in a textbook? And then here, that's really good too. But, it was almost like something happened, something different happened.
So let me see. So for the Na, for the sodium, potassium, ATP, ACE, that's the first one, it was my fault because I mislabeled the proteins, and so I added a different protein to it, which I think, if I'm being honest, it might have been the AP2. Now that I'm looking at it. The PAP2 actually. Now that I'm looking at it, and so we had to do a second try of that, which is this.
So Kai brought up a really good point. She said she made a mistake. Did anybody else make a mistake this summer? Or is she the only one? Okay. All right, last but not least, we have Eric. And he is going to be introduced by his mentor, Dr. Delgado.
All right, so first, I'd like to thank the organizing committee for allowing me to participate in the program again this year, because with that, I wouldn't have had Eric in my lab for the second year in a row. I drug him back. So Eric and I got along pretty well, probably because he's originally from New York, like I am. But now he's from Pittsburgh and he hails from Cranberry Township, where he went to Seneca Valley. And I say went because Eric just graduated this year, and he'll be starting at Johns Hopkins in the fall, where his older brother, another alumni of this program, currently attends. In the lab, Eric is very patient, especially with me. In terms of especially making mistakes in the lab, and some of these mistakes come from me. And these particularly involve making drug dilutions and drug calculations, where Eric tends to pick up on all of my mistakes and my miscalculations. He's a quick learner, and he's he's very adept at taking work and pushing it forward on his own independently with very little supervision. And also, you should see his notebook. It's like a class A example of like how a notebook should be kept. So I greatly really appreciated having Eric in my lab for the past 2 years, and hopefully I can wrangle him back to coming to my lab to do another summer undergraduate research experience in my new lab. And with that, I'm happy to have Eric come talk to you about what he's been doing for the past couple of weeks. That's a good question. I don't think I do.
See, I'm making mistakes all the time.
Cool. So as Evan introduced, I'm Eric, and my project was working on if epigenetic inhibitors can promote gene expression in hepatocellular carcinoma. All right, so hepatocarcinoma, or HCC, is a multi-step process and usually derived from chronic injuries, such as from drinking too much or hepatitis. And it's the most common type of primary liver cancer. Okay. So HCC presents a significant public health burden because in 2022, what's going on? Oh, there we go. In 2022, there was about 40,000 new cases of HCC, about 30,000 of them resulting in death. This makes HCC the sixth most common cause of cancer-related death in the United States. And unfortunately, this trend is expected to increase throughout the years. So regarding clinically approved first-line defenses, sorafenib, which you may have heard Alberto mention earlier, was the main first line since the early 2000s. But in 2018, there's been a new combo of atezo and bev that has been utilized to improve survival rates in comparison to sorafenib. This combination has been approved for first-line defense. And you can see that, oops, I can use a laser pointer. It can improve it in comparison to just sorafenib alone. However, due to tumor heterogeneity, this response only affects about 30% of patients, and for about the other 2/3, it doesn't do anything at all. And that's not particularly good. This leads me to my research question. Is there a strategy that we can use to improve the efficacy of immunotherapy in HCC cells? Oh, and just HCC rather. My project takes inspiration from a study performed on ovarian cancer. Like liver cancer, ovarian cancer is also notoriously difficult to treat with immunotherapy. From the study, they performed using a HDAC inhibitor as well as a DNA methyltransferase, or DMNT. Together, these two caused an increase in immune-related gene expression. So based on the results of that study, my mentor and I tried to formulate an experiment with this for HCC. And you can see the results right there. After some research, we found that this study that characterized the HDAC RNA expression in both human and mouse models. This top is human cell lines, and this bottom is mouse cell lines. And across the board, we can see that HDAC RNA expression is elevated in HCC cancer samples. This group also compared just the overall HDAC expression to from cancer samples from the Cancer Genome Atlas. And from this, they found that the expression of these HDACs was also correlated to a decrease in patient survival rate. About 10 years ago, there was a clinical trial trying to use belinostat, which is the HDAC inhibitor I'm working with this summer. It's been shown to be well tolerated in patients, but due to underwhelming survival benefits, it didn't move into phase 3 or clinical practice. Belinostat is a pan-HDAC inhibitor, which means it inhibits HDACs across the board. It doesn't target a specific one. And as such, it should increase the degree of acetylation in chromatin, which should enhance the rate of gene expression. Based on this, we formulated our experiment if we can utilize belinostat to increase gene expression in immune-related genes. So this is just some data that I took from last year's progress. As you can see, we used HEPA-1/6 cells, which is a type of cancerous mouse cell line, of mouse liver cell line. And in a 24-hour treatment, you can see that at a higher 100 nanomolar doses, there is significant increases in gene expression. However, this was not replicated in the 48-hour or the 72-hour. So following up with these results, we decided to expand further using new cell lines. I worked with HEPA-1/6 at the very beginning to basically expand out the types of genes that I want to test, and also worked with AML12 and primary human and mouse hepatocytes. With this, we treated them with a dose escalation study, which is basically just moving from 0 up to a higher dose, such as like 800 nanomolars, just to see the gene expression as the dose increases. We also used a sequential treatment study in which we treated the same cells multiple times using fresh belinostat and 5-AZA every day. After these treatments, we used a luminescence-based assay to measure ATP and the cell viability, or how well these cells are thriving, like if they're living and producing like ATP. We also used qPCR to measure the gene expression of the immune-related genes that I'm about to show up there. To begin, we're curious about the expression of a few other immune-related genes, such as the interferon gamma receptors and IL-6. You can see how in the HEPA-1/6 cells, this expression is elevated in the 24 hours time point, but not in the 48 and 72 hours, which corresponds with what I had received last year. Interferon gamma and the interferon gamma receptors act as a lock and a key. When these two meet, they trigger a reaction that recruits more immune cells, which also brings in more interferon gamma. And this essentially creates a reaction, a chain reaction rather, that amplifies immune response. After pinpointing some genes of interest, we decided to move on with AML12, which is a type of primary mouse liver cell line. We repeated the dose escalation study with this, and as you can see, the cell viability is not significantly altered. However, the gene expression is essentially shut down for most of the concentrations that we used. In the sequential treatment study, while we did see that the cell viability did not remain, was not changed in a statistically significant way, the gene expressions for the interferon gamma receptors, CXCL1 and CCL20, were improved by a significant margin. Next, we moved on to primary mouse hepatocytes. As AML12 it's derived from the primary mouse hepatocytes, but it may have changes in between. So we harvested these directly from a mouse through a perfusion, and we just started culturing them. As you can see in the dose escalation curve, I used a lot of different genes, the interferon gamma receptors, the CXCLs and the CCLs. And overall, we see either a downward trend in gene expression as the dose of belinostat increases, or there's no significant change. And the cell viability does not change either. However, in the sequential treatments with these cells, we found that while the cell viability did not, while the cell viability does not change a lot, we also begin to see decreases in interferon gamma receptors, while there's an increase in the CXCL1 still. Let's not really move forward into the slide.
Sometimes if you just close. Where's the?
Okay, thank you. So after the primary mouse hepatocytes, we moved on to using primary human hepatocytes and see if these results were transferred across from mouse to humans. Unlike in the mouse models, we saw a significant decrease in cell viability, and we still saw a decrease in the interferon gamma receptors 1 and 2. However, in the sequential dose study for the human hepatocytes, we saw a substantial increase in the gene expressions. These two bars right here are both about 1, and those are our controls. So seeing a massive increase about 50 times for the intergamma receptors could prove promising. And the cell viability is still not changed in a significant way. So overall, we found that the belinostat and potentially other HDAC inhibitors have a more immediate response in cancer cells as opposed to normal hepatocytes, as shown in the HEPA-1/6 result in comparison to these normal hepatocytes. We also found that this combination was likely, appears to be non-toxic to liver cells. In the future, we wish to see if these results will be replicated in cancerous humans liver cells. Originally, we had planned to use these cells, but due to suboptimal culturing conditions, they did not survive to see the treatment process. We also want to see if these elevated expressions was correlated to proteins, because that's something we did not look into. And the presence of these proteins would confirm that the gene expressions was in fact working to help recruit immune-related immune cells. And finally, we wanted to see if these results would be shown in just other immune-related genes. The genes that I worked with were shown to be part of both human and mouse liver cells, but at the same time, these were still hand-picked and weren't necessarily guaranteed to be shown in other immune-related genes. And with that, I'd like to thank everyone who has helped me with this project, specifically Evan, who has mentored me across these 2 years, as well as other members of the lab like Anisha, who has helped me throughout my project. Many thanks to the directors of Hillman Academy, as well as Dr. Duncan and Dr. Gao, and just to everyone who has helped make this opportunity possible. Thank you.
Those are really cool results, and I like the way you put them all together with the different cell types. One of the things I know your lab is really exceptional at is mining these sort of public databases like the TCGA database, where you can look at all these patients who have got different kinds of tumors and get different kinds of treatments. And so have you thought about learning how to use the TCGA database or equivalent ones to see patients who have gotten certain kinds of these inhibitors, and to see what happens to their target genes of interest? Basically, can you do the experiment in humans by mining those databases?
It's a really good question. That's something I haven't quite looked into, but that would be a really great resource to use in terms of like just seeing if these inhibition, like HDAC inhibitions, would affect the liver cancer.
You did some of these studies where you did like 2 treatments, 3 treatments in a row. We saw a lot of changes with the gene expression. I wonder if you have any comments and stuff or thoughts on kind of like why you think like the 2 doses or 3 doses had such a dramatic difference compared to like just like the 1 dose. And I think it was your last graph I saw was like this one right here. You did a 1x and it's like 60-fold change, and then you did 2x and it was like 1.5.
To be honest, I'm not quite sure about why specifically. We think that has something to do with like just how the cell responds to the the opening of the chromatin. because this is also treated with a DMNT, which helps prevent the like closing of the chromatin. So this is just like constantly like opened. But in the 2x treatments, we are like removing the media from the cells again. So like anything that's shown in the 1x is now transferred over to 2x. So the 2x is only like the ATP, sorry, not the ATP, the RNA that's produced during the like after the 24 hours of initial treatment. So that could be why it's not like transferred over from the 24-hour to the 48-hour. Thank you.
Well, that concludes, I think, 7 really outstanding presentations. So just another round of applause to all the presenters. And, I want to give a special thanks to all the mentors who came and worked with the students throughout the summer. And I think I want to really thank the students for their outstanding job, and thank you to the parents for trusting us with the kids all summer long. We appreciate that responsibility and take it very seriously, which I think you saw. So do you have any comments? So logistically, where we're at now is I am told there should be lunch right here where the other stuff was. You can grab that and bring it in here. For those of you who are going to go over to the assembly building, that closing ceremony starts at 2 o'clock. If you're driving, you can go over there whenever you like. If anybody needs the shuttle, I'm told it leaves off of Lothrop, which is right over here at 12:55. Is anybody riding the shuttle? Okay, all right. Well, thank you guys for coming. And oh, lastly, one opportunity we have is the Department of Pathology does an annual retreat every May. And one of the things that we do is a big poster session where we invite the trainees, who are graduate students and residents and postdocs, to give talks and to present posters. So all of the students in the pathobiology group here, if you guys would like to come back in May and attend our poster session, you're welcome to do that. And I'll send out information. So you could basically submit your abstract, and then you'd have to take all this stuff and put it on a poster. But there's some opportunities to do that if you'd like. So we can talk about that later on. Thank you, guys.